Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Diversity of Plants Zone. In this channel, you can easily find the amazing videos which are related to plants and their diversity throughout the world. Before starting my today's topic, I request you all to please subscribe to my YouTube channel. So, today's topic of my video is about three-domain classification system. So, let's start the video. First of all, I will discuss about the domain. In biological taxonomy, a domain, Latin, is also known as the regio, also known as the dominion, superkingdom, realm, or empire, is the highest taxonomic rank of organisms in the three-domain system of taxonomy, devised by Carl Woese et al. In 1990. According to this system, the tree of life consists of three domains, which are Archaea, Bacteria, and Eukarya. The first two are all prokaryotic microorganisms, or mostly single-celled organisms whose cells have a distorted or non-membrane bound nucleus. All life that has a cell nucleus and eukaryotic membrane bound organelles is included into the Eukarya. Non-cellular life is not included in this system. Alternatives to the three-domain system include the earlier two empire system, with the empires Prokaryota and Eukaryota, and the Eocene hypothesis, with the two domains of Bacteria and Archaea and Eukarya included within Archaea. So this is the hierarchy of biological classification's 8 major taxonomic ranks. So life is divided into domains, which are subdivided into further groups. Intermediate, minor rankings are not shown. So this hierarchy of biological classification includes life, then comes domain, kingdom, phylum, class, order, family, genus, and species. Next, I am going to discuss about the terminology. The term domain was proposed by the Carl Woese, Otto Candler, and Mark Willis in 1990 in a three-domain system. This term represents a synonym for the category of dominion. Latin, it is called as the dominium, introduced by Moore in 1974. Next, I am going to discuss about the three-domain system. The three-domain system is a biological classification introduced by Carl Woese et al. In 1990 that divides the cellular life forms into Archaea, Bacteria, and Eukaryote domains. The key difference from the earlier classifications is the splitting of Archaea from Bacteria. So these are the three domains. This is a phylogenetic tree which is based on ribosomal RNA data and emphasizing the separation of Bacteria, Archaea, and Eukaryotes as proposed by the Carl Woese and George E. Fox et al. In the 1990. So first is the domain Bacteria, and Bacteria includes Chloroflaxi, Gram-positives, Spirochetes, Proteobacteria, Cyanobacteria, and Plantomyces, Bacteroides, Cytophaga, Thermophiles, and Aquifex. On the other hand, the Archaea includes Haloarchaea, Methanosarcina, Methanobacterium, Methanococcus, Thermococcus cellular, Thermoproteus, Pyrodicticum. Next is the domain Eukaryota, which includes Entamoeba, slime molds, animals, fungi, plants, ciliates, flagellates, trichomonads, and microsporidia, and then comes diplomonads, as shown into the figure. The three-domain system is a biological classification introduced by Carl Woese in 1990 that emphasizes his separation of prokaryotes into two groups, originally called Eukarya and Archaea Bacteria. Was argued that based on differences in 16 subunit ribosomal RNA genes, these two groups and eukaryotes each arose separately from an ancestral progeny with poorly developed genetic machinery. To reflect these primary lines of descent, he treated each as a domain, divided into several different kingdoms. The groups were also renamed the Bacteria, Archaea and Eukarya, further emphasizing the separate identity of the two prokaryote groups. Although the three-domain system was quickly adopted by most molecular systematists, biologists like Ernst Mayr criticized him for overemphasizing the uniqueness of the Archaea bacteria and ignoring strong genetic similarities between the groups. Subsequent studies have confirmed that the Archaea are unusual in the composition of their cell membrane and structure of their flagella. Other significant differences include archaeal systems for DNA replication and transcription, which bear a distinct similarity to those found in eukaryotes. For instance, archaeal RNA polymerase consists of up to 14 subunits, whereas the most bacterial RNA polymerases have only 4 subunits. Analysis of these subunits suggests that they are more closely related to those found in eukaryotes. Also, the Archaea produce a number of DNA binding proteins with similarity to eukaryotic histones. Nevertheless, a minority viewpoint suggests retaining the older two empire system, Prokaryota and Eukaryota, and using the word bacterium in its earlier meaning of prokaryote. Which system is preferable? That depends partially on the relationships of organisms in the question. Although the prokaryote hypothesis is discredited, molecular trees tend to group living things into 3 domains, with the eukaryotes placed beside or within Archaea, and Eubacteria forming a separate branch. However, it has been suggested that this is an artifact of long branch attraction, and that the root may instead belong among the Eubacteria, in which case many Eubacterial lines diverged before the Archaea Bacteria did. In 2006, the discovery of unique properties of the virus started a discussion about the classification of certain viruses as a 4th domain of life. Next, I am going to discuss about the background was argued on the basis of differences in 16 subunit ribosomal RNA genes that bacteria, archaea, and eukaryotes each arose separately from an ancestor with poorly developed genetic machinery, often called as a progenote. To reflect these primary lines of descent, he treated each as a domain, divided into several different kingdoms. Originally, his split of the prokaryotes was into Eubacteria, now Bacteria, and Archaea Bacteria, now Archaea. Woese initially used the term kingdom to refer to the 3 primary phylogenetic groupings, and this nomenclature was widely used until the term domain was adopted in 1990. Acceptance of the validity of Woese's phylogenetically valid classification was a slow process. Prominent biologists including Salvador Luria and Ernst Mayr objected to his division of the prokaryotes. Not all the criticism of him was restricted to the scientific level. A decade of labor-intensive oligonucleotide cataloging left him with a reputation as a crank, and Woese would go on to be dubbed microbiology's SCAD revolutionary by a news article printed into the journal Science. The growing amount of sporting data led the scientific community to accept the Archaea by the mid-1980s. Today, few scientists cling to the idea of a unified prokarya. So next is the most important topic. It is about the characteristics of the three domains. Each of these three domains contains a unique ribosomal RNA, and this forms the basis of the three-domain system. While the presence of a nuclear membrane differentiates the Eukarya from the Archaea and Bacteria, both of which lack a nuclear membrane and distinct biochemical and RNA markers differentiate the Archaea and Bacteria from each other. Next is the question, how are three domains related? Comparing ribosomal RNA-based sequences, Woese and his colleagues also showed that organisms belonging to Eukarya are more similar to Archaea than they are to bacteria. So figure below is a phylogenetic tree which is based on their analysis. This tree places Archaea and Eukarya in the same clade. It represents the hypothesis that Archaea and Eukarya shared a more recent common ancestor with each other than either shared with bacteria. This phylogenetic tree is based on the comparisons of ribosomal RNA-based sequences among living organisms. The tree divides all the organisms into three domains, which are bacteria, Archaea, and Eukarya. Humans and other animals belonging to the Eukarya domain. From this tree, organisms that make up the domain Eukarya appear to have shared a more recent common ancestor with Archaea than bacteria. The results of a study published in 2007 seem to conflict with this hypothesis. Comparing DNA-based sequences, the 2007 study suggested that the domain Archaea may be older than either bacteria or Eukarya. That would make Archaea the most ancient group of organisms on Earth. It is not yet known which if either hypothesis is correct. Scientists need to learn more about Archaea and their relationships with other organisms to resolve these questions. So this is again a speculatively rooted tree for RNA genes showing the major branches bacteria, Archaea and Eukaryota. So this is the first domain which includes is bacteria, which includes Chlorophyllaceae, gram-positive, Spirochetes, Proteobacteria, Cyanobacteria, and Planktomyces and Bacteroides, Cytophaga, Thermophiles, and Aquifax. Next domain is Archaea, which includes Haloarchaea, Methanosarcina, Methanobacterium, Methanococcus, Thermococcus cellular, Thermoproteus, and Pyrodicticum. And the last is the Eukaryota. It includes entamoeba, slime molds, animals, fungi, plants, ciliates, flagellates, trichomonads, and microsporidia and diplomonads, as shown into the figure. So next I am going to discuss one by one. First is the Archaea. Archaea are prokaryotic cells typically characterized by membrane lipids that are branched hydrocarbon chains attached to the glycerol by ether linkages. The presence of these ether linkages in Archaea adds to their ability to withstand the extreme temperatures and highly acidic conditions, but many Archaea live in the mild environments. Halophiles are organisms that thrive in highly salty environments, and hyperthermophiles, organisms that thrive in extremely hot environments, are examples of Archaea. Archaea involved many cell sizes, but all are relatively small. Their size ranges from 0.1 micrometer to 15 micrometer diameter and up to 200 micrometer long. They are about the size of bacteria or similar in the size to the mitochondria, which is found in eukaryotic cells. Members of the genus Thermoplasma are the smallest of the Archaea. So next comes bacteria. Even though bacteria are prokaryotic cells just like Archaea, their membranes are made up of phospholipid bilayers. So cyanobacteria and mycoplasmas are the two examples of bacteria. They characteristically do not have the ether linkages like Archaea, and they are grouped into a different category and hence a different domain. There is a great deal of diversity in this domain. Confounded by the diversity and horizontal gene transfer, it is next to impossible to determine how many species of bacteria exist on the planet, or to organize them in a tree structure without cross connections between branches. Next comes Eukarya. So members of the domain Eukarya called eukaryotes and have membrane-bound organelles, including a nucleus containing genetic material, and are represented by 5 kingdoms, which are Planty, Protozoa, Animalia, Chromista, and Fungi. So this is a figure over here, the 3 domains tree and eocyte hypothesis. 2 domains tree 2008. First of all, I will discuss about the 2 domains tree. 2 domains tree have 2 domains, bacteria, Eukarya, okay, and this is Archaea, and Archaea includes some subdomains in it, which are Urakuta. This is Urakuta, and then comes Asgard. Asgard is also known as the Asgard Archaeota. Asgard Archaeota is a proposed subphylum which is consisting of a group of Archaea that includes Lochia Archaeota, and next is the Thorakuta and Orden Archaeota, and last is the Heimdall Archaeota. On the other hand, this is next TAC, T-A-C-K, TAC. So TAC is also a group of Archaea, which is acronym for the Thermophiles, Eukaryote, and Eukaryote and Eukaryote, the first groups which are discovered. So the tag includes Eukaryote, Eukaryote, and Thermophiles, and the last is the Proteobacteria. This is a 2-domain tree, and the next is the 3-domain tree. This is the Carl Woese 3-domain tree, and it includes 3 domains: bacteria, Archaea, and Eukarya. And Archaea includes 2 subdivisions, which are Urakuta and Eukaryote. This is as shown into the figure. This is a phylogenetic tree which is showing the relationships between the eukaryotes and other life forms. 2006, eukaryotes are colored red, Archaea green, and bacteria blue. So first of all, I will discuss about bacteria. Bacteria, gram-positives and Chlamydiae, green non-sulfur bacteria, Actinobacteria, planktomycetes, Spirochetes, and Fusobacteria, Cyanobacteria, blue-green algae, which is also known as blue-green algae, and thermophilic sulfate reducers, Acetophiles, and Proteobacteria. So the next is the Archaea. Archaea includes 3 subdivisions, and which are Urakuta, Nanoarchaeota, and Eukaryote, and the next is the Eukaryotes. So eukaryotes includes protozoa, algae, plants, live molds, animals, and fungi. All these are shown into the figure. So next I am going to discuss about classification. The 3-domain system adds a level of classification, the domains above the kingdoms present in the previously used 5 or 6 kingdom systems. This classification system recognizes the fundamental divide between the 2 prokaryotic groups insofar as Archaea appear to be more closely related to eukaryotes than they are to other prokaryotes, bacteria-like organisms with no cell nucleus. This system sorts the previously known kingdoms into these 3 domains, which are Archaea, Bacteria, and Eukarya. First of all, I will discuss about domain Archaea. The Archaea are prokaryotic with no nuclear membrane but distinct biochemistry and RNA markers from bacteria. The Archaeans possesses unique ancient evolutionary history, for which they are considered some of the oldest species of organisms on Earth, most notably their diverse exotic metabolisms. Some examples of archaeal organisms are methanogens, which reproduce the gas which is called methane, halophiles, which live into the salty water. Next is thermoacetophiles, which thrive in acidic high temperature water. Next is domain bacteria. The bacteria are also prokaryotic. Their domain consists of cells with bacterial ribosomal RNA, no nuclear membrane, and whose membranes possesses primarily diacyl, glycerol, diester, lipids. Traditionally classified as bacteria, many thrive in the same environments as favored by humans and were the first prokaryotes discovered. They were briefly called as the Eukarya or true bacteria, when the Archaea were the first recognized as a distinct clade. Most non-pathogenic prokaryotic organisms belong to bacteria. For that reason, and because the Archaea are typically difficult to grow in laboratories, the bacteria are currently studied more extensively than Archaea. Some examples of bacteria include Cyanobacteria, photosynthesizing bacteria that are related to the chloroplasts of the eukaryotic plants and algae. Next is Spirochetes, which is also called as gram-negative bacteria, that include those causing syphilis and Lyme disease. Next comes Actinobacteria, and this is also known as gram-positive bacteria, including Bifidobacterium animalis, which is present into the human large intestine. Next comes Domain Eukarya. Eukarya are organisms whose cells contain membrane-bound nucleus. They include many large single-celled organisms and are non-microscopic organisms. A partial list of eukaryotic organisms include kingdom fungi or fungi, and it includes Saccharomycotina, which include true yeasts. Then comes Basidiomycota, includes mushrooms. Then comes kingdom Planty or plants, which includes Bryophyta, mosses, and Magnoliophyta, flowering plants. Next kingdom is kingdom Animalia or animals, which includes Chordata, includes vertebrates as a subphylum. So next I am going to discuss about the niches. Each of the three cell types tend to be fit into the recurring specialities or roles. So bacteria tend to be the most prolific reproducers, at least in moderate environments. Archaeans tend to adapt quickly to extreme environments such as high temperatures, high acids, and high sulfur, etc. This includes adopting to use a wide variety of food sources. Eukaryotes are the most flexible with regard to forming cooperative colonies, such as in the multicellular organisms including humans. In fact, the structure of a eukaryote is likely to have derived from a joining of different cell types, forming organelles. So, Prokaryotic Myogenensis, which is also called as Incertaceous, is a single-celled organism known to be a unique example. This organism appears to be a life form distinct from the prokaryotes and eukaryotes, with features of both. Next are alternatives. I am going to discuss about alternatives. Parts of the three-domain theory have been challenged by scientists including Ernst Mayr and Thomas Cavalier Smith. In particular, one of them argues that the primary division within prokaryotes should be among those surrounded by a single membrane monoderm, including gram-positive bacteria and Archaebacteria, and those with an inner and outer cell membrane diaderm, including gram-negative bacteria. He claims that sequences of features and phylogenes from some highly conserved proteins are inconsistent with the three-domain theory and that it should be abandoned despite its widespread acceptance. Recent work has proposed that Eukarya may have actually branched off from the Domain Archaea. According to the Spank et al., Eukaryote forms a monophyletic group with eukaryotes in phylogenomic analysis. The associated genomes also encode an expanded repertoire of eukaryotic signature proteins that are suggestive of sophisticated membrane remodeling capabilities. This work suggests a two-domain system as opposed to the near universally adopted three-domain system. So this is a figure. These are the variants of the three-domain system. First variant includes Bacteria, Archaea, Eukaryota. Second variant includes Bacteria, Archaea, Eukaryota. Third variants include Bacteria, Archaea, Eukaryota. Fourth variants also include Bacteria, Archaea, Eukaryota. And this is fifth variant, which includes Archaea, Eukaryota and Bacteria as shown into figure. So next is the topic about the exclusion of the viruses and parents. The three-domain system doesn't include any form of non-cellular life. So Stefan Urakuta proposed a five-domain system in 2012 and adding the Protiobiota, acellular and without nucleic acid, and Virusobiota, acellular but with nucleic acid to the traditional three domains. This is the alternative classification as shown into the figure. So in this table, first I will discuss about taxonomical root node. Taxonomical root node, then comes two superdomains, controversial, and two empires, three domains, five kingdoms, six kingdoms, and eocide hypothesis. So first is the taxonomic root node, which includes biota and vitae or life. Two superdomains controversial are Acetophiles and Aphanobionta, non-cellular life. And the next is the Cytota, which is cellular life. Two empires includes Prokaryota, which is also known as Prokarya and Monera, and the next is Eukaryota, Eukarya. Three domains include Bacteria, Archaea, and Eukarya. Five kingdoms include Virusobiota, which includes viruses and viroids. Next is the Cyanobacteria, which includes prions, and Bacteria and Archaea, and Eukarya. And the next is the six kingdoms. The six kingdoms are Eubacteria, Archaebacteria, Protista, Fungi, Planty, and Animalia. Next is eocide hypothesis, which includes Bacteria, Archaea, including Eukaryotes. Alternative classification of life include the two empire system or super domain system, with the top level groupings of Prokaryota or Monera and Eukaryota. The eocide hypothesis was proposed by the James A. Lake et al. In 1984, which posits two domains, Bacteria and Archaea, with Eukaryota included in Archaea. Next I am going to discuss about classification, the three-domain system. The Earth is about 4.6 billion years old, and microbial life is thought to have first appeared between 3.8 and 3.9 billion years ago. In fact, 80% of the Earth's history was exclusively microbial life. Microbial life is still the dominant life form on Earth. It has been estimated that the total number of microbial cells on Earth on the order of 2.5 into 10 raised power 30 cells, making it the major function of biomass on the planet. Phylogeny refers to the evolutionary relationships between organisms. The three-domain system proposed by Woese and others is an evolutionary model of phylogeny based on differences in the sequences of nucleotides in the cell's ribosomal RNAs, as well as the cell's membrane lipid structure, its sensitivity to antibiotics. Comparing ribosomal RNA structure is especially useful because ribosomal RNA molecules throughout the nature carry out the same function. Their structure changes very little over time. Therefore, the similarities and dissimilarities in ribosomal RNA nucleotide sequences are a good indication of how related or unrelated different cells and organisms are. There are various hypotheses as to the origin of prokaryotic and eukaryotic cells. Because all the cells are similar in nature, it is generally thought that all the cells came from a common ancestor, and cell termed the last universal common ancestor. This is also known as LUCA. These LUCAs eventually evolved into three different cell types, and each representing a domain. The three domains are Archaea, the bacteria, and Eukarya. The more recently, various Fusion hypotheses have begun to dominate the literature. One purpose is that the diploid or two-end nature of the eukaryotic genome occurred after the fusion of two haploid or one-end prokaryotic cells. Others purpose that the domains Archaea and Eukarya emerged from a common and archaeal eukaryotic ancestor that itself emerged from a member of the domain bacteria. Some of the evidence behind this hypothesis is based on the superphylum of bacteria, which is called PVC, members of which share some characteristics with both Archaea and Eukaryotes. There is a growing evidence that Eukaryotes may have originated within a subset of Archaea. In any event, it is accepted today that there are three distinct domains of organisms in nature, which are bacteria, Archaea, Eukarya. A description of three domains follows. So first is the Archaea, which is also known as Archaea Bacteria. The Archaea possesses the following characteristics, which are: Archaea are prokaryotic cells. Unlike bacteria and the Eukarya, the Archaea have membranes composed of branched hydrocarbon chains, many also containing rings within their hydrocarbon chains, attached to the glycerol by ether linkages. The cell walls of Archaea contain no peptidoglycan. Like the Archaea are not sensitive to some antibiotics that affect the bacteria, but are sensitive to some antibiotics that affect the Eukarya. Archaea contain ribosomal RNA that is unique to the Archaea, as indicated by the presence molecular regions distinctly different from the ribosomal RNA of bacteria and Eukarya. So these are the membrane lipids of Archaea Bacteria and Eukarya. The bacteria and the Eukarya have membranes composed of the unbranched fatty acid chains, which are attached to the glycerol by ester linkages. So this is the membrane of bacteria and Eukarya. So these unbranched fatty acid chains are attached to the glycerol. This is glycerol, and these are attached to it by the help of the ester linkages as shown into the figure. And the next, the Archaea have membranes composed of branched hydrocarbon chains. These are all branched hydrocarbon chains, you can see, which are attached to the glycerol. So these branched hydrocarbon chains are attached to the glycerol by the help of the ether linkages. This is ether linkages as shown into the figure. Archaea often live in extreme environments and include methanogens, extreme halophiles, and hyperthermophiles. One reason for this is that the ether containing linkages in the Archaea membranes is more stable than the ester containing linkages in the bacteria and Eukarya, and are better able to withstand the higher temperatures and stronger acid concentrations. So next is the bacteria, which is also known as Eubacteria. Bacteria, also known as Eubacteria or true bacteria. are prokaryotic cells that are common in human daily life, encountered many more times than the Archaea bacteria. Eubacteria can be found almost everywhere and kill thousands upon the thousands of people each year, but also serve as antibiotics producers and food digesters in our stomachs. The bacteria possesses the following characteristics. First, bacteria are prokaryotic cells. Like the Eukarya, they have membranes which are composed of unbranched fatty acid chains attached to the glycerol by ester linkages. The cell walls of bacteria, unlike the Archaea and the Eukarya, contain the peptidoglycan. Bacteria are sensitive to traditional antibacterial antibiotics but are resistant to most antibiotics that affect Eukarya. Bacteria contain ribosomal RNA that is unique to bacteria as indicated by the presence of molecular regions distinctly different from the ribosomal RNA of Archaea and Eukarya. Bacteria include many mycoplasmas, Cyanobacteria, gram-positive bacteria and gram-negative bacteria. The Eukarya. Next is the Eukarya, which is also called as Eukaryotes. The Eukarya, also spelled as Eukarya, possesses the following characteristics, which are: Eukarya have eukaryotic cells. Like the bacteria, they have the membranes composed of unbranched fatty acid chains attached to the glycerol by ester linkages. Not all the Eukarya possesses cells with a cell wall, but for those Eukarya having a cell wall, that wall contains no peptidoglycan. Eukarya are resistant to the traditional antibacterial antibiotics but are sensitive to the most antibiotics that affect eukaryotic cells. Eukarya contain ribosomal RNA that is unique to the Eukarya as indicated by the presence of molecular regions distinctly different from the ribosomal RNA of Archaea and bacteria. The Eukarya are subdivided into following four kingdoms, which are Protista Kingdom. Protista are simple, predominantly unicellular eukaryotic organisms. Examples include slime molds, euglenoids, algae and protozoans. Next is the Fungi Kingdom. So fungi are unicellular or multicellular organisms with eukaryotic cell types. The cells have cell walls but are not organized into tissues. They do not carry out photosynthesis and obtain nutrients through absorption. Examples include sac fungi, club fungi, yeast and molds. Next is Planty Kingdom. So plants are multicellular organisms composed of eukaryotic cells. The cells are organized into tissues and have cell walls. They obtain nutrients by photosynthesis and absorption. Examples include mosses, ferns, conifers and flowering plants. Next is the Animalia Kingdom. So animals are multicellular organisms composed of eukaryotic cells. The cells are organized into tissues and lack the cell walls. They do not carry out the photosynthesis and obtain nutrients primarily by ingestion. Examples include sponges, worms, insects and vertebrates. It is used to be thought that the changes that allow microorganisms to adopt to new environments or alter their virulence capabilities was a relatively slow process, occurring within an organism primarily through mutations, chromosomal rearrangements, gene deletions and gene duplications. Those changes would then be passed on to that microbe's progeny and natural selection would occur. This gene transfer from a parent to organism to its offspring is called the vertical gene transmission. It is now known that microbial genes are transferred not only vertically from a parent organism to its progeny but also horizontally to the relatives that are only distantly related, for example, other species and other genera. This latter process is known as the horizontal gene transfer. Through mechanisms such as the transformation, transduction and conjugation, genetic elements such as plasmids and transposins and integrants and even chromosomal DNA can readily be spread from one microorganism to another. As a result, the old three-branch tree of life in regard to microorganisms now appear to be more of a net of life. Microbes are known to live in remarkably diverse environments, many of which are extremely harsh. This amazing and rapid adaptability is a result of their ability to quickly modify their repertoire of protein functions by modifying, gaining, or losing their genes. This gene expansion predominantly takes place by horizontal transfer. So this is a further explanation about the Carl Woese classification. The three-domain system was first introduced by the Carl Woese in 1990. That is why it is called Carl Woese classification. This classification system is also known as the Six Kingdoms and Three Domains classification system because it divides the life forms into three domains and six kingdoms. The three domains of Carl Woese classification system includes Archaea Bacteria, Eukaryote, and six kingdoms are Archaea Bacteria, Ancient Bacteria, Eukaryotic, True Bacteria, Protista, Fungi, Planty, and Animalia. This classification system divides the life form based on the differences in the 16 subunit ribosomal RNA structure and as well as the cell membrane lipid structure and its sensitivity to antibiotics. The main difference from earlier classification systems is the splitting of archaea from bacteria. The evaluating ribosomal RNA structure is very helpful. As a result of ribosomal RNA molecules throughout the nature perform the identical function, their structure modifications vary little over time. Subsequently, similarities and dissimilarities in ribosomal RNA nucleotide sequences are a very good indication of how the associated or unrelated completely different cells and organisms are. In this classification, Carl Woese uses the 16 subunit ribosomal RNA as a chronometer. Because of it is universally distributed means its presence in all species. It is functionally similar in all organisms. It can change its sequence slowly. Its sequence can be aligned or matched up between two organisms. Carl Woese classification is also known as the three-domain system. This three-kingdom system was first proposed by an American microbiologist and biophysicist called Richard Woese in 1990. This classification system divides the life forms into three domains and six kingdoms. That is why it is called the Six Kingdoms and Three Domains classification. The three domains are Archaea Bacteria, Eukaryote, and six kingdoms are Archaea Bacteria, Ancient Bacteria, Eukarya, which is also called True Bacteria, Protista, Fungi, Planty, Animalia. Woese classified them based on their differences in the 16 subunit ribosomal RNA structure. In the 1987, Carl Woese of the University of the Illinois, USA proposed a phylogenetic classification of prokaryotic species which was based on the differences in their nucleotide sequence of the 16 subunit ribosomal RNA molecules. Later, he concludes that the 16 subunit ribosomal RNA can be used for comparative analysis between prokaryotic and eukaryotic species. This provides a tree of relatedness based on the common ancestry or genealogy, because both prokaryotic and eukaryotic cells contain the small subunit ribosomal RNA, SSU ribosomal RNA. Carl Woese used the ribosomal RNA as an evolutionary chronometer, an evolutionary time clock. So now the question, why is ribosomal RNA a good chronometer? Ribosomal RNA is universally distributed. All the species have ribosomal RNA. So ribosomal RNA is functionally similar in different organisms. It participates in the synthesis of protein. Ribosomal RNA changes its sequence slowly. It means it is optimum for looking across the periods of time, looking across long periods of time. And ribosomal RNA sequences can be aligned or matched up between two organisms. So, domains of the Carl Woese classification. Carl Woese classification is made up of three domains such as Domain Archaea, Domain Bacteria, and Domain Eukarya. I will discuss it one by one. So first is the Domain Archaea. The Archaea domain includes all prokaryotic cells. They lack the nuclear membrane and have distinct biochemistry, contain RNA markers from bacterial cells. The Archaea are considered as the oldest species of organisms on Earth, and they can survive in extreme harsh environments that differentiated them from other domains. The cell walls of Archaea lacks the peptidoglycan and also contains ether linkages in their membranes. Archaea has three phyla, such as the Eukaryote. They can survive at extremely high temperatures and extremely low temperatures. Next is the Urakuta, which is also called Eukaryote. Some of them are known as extremely halophiles, which can inhibit highly saline environments. Next is the Prokaryote. It includes all those species which were found in a single hot spring, like the obsidian pool present in the Yellowstone National Park, USA. They are prokaryotic cells with no nuclear membrane. Archaea has a distinct biochemistry and RNA markers from bacteria. They have isoprenoid, glycerol, diether or diglycerol, tetraether lipids in their membranes. They contain archaeal ribosomal RNA. Archaea lacks the peptidoglycan in their cell wall. They are not sensitive to those antibiotics. Those are used for the treatment of bacteria, but they are sensitive to some antibiotics that are affecting the Eukarya. Archaea contain ether linkages in their membrane, which is formed between branched hydrocarbon chains and glycerol. Archaea lacks the peptidoglycan in their cell wall, so they can thrive in extreme environments such as high temperature, low temperatures, stronger acidic concentrations, etc., because they have a more stable ether linkage as compared to the ester linkage containing linkages in the bacteria and Eukarya. Next, I am going to discuss about the phyla of Archaea. Archaea has three major phyla, such as Eukaryote. This phylum is consisting of those microorganisms which are lived in extremely high temperature and extremely low temperature. Some of the Eukaryote can thrive above the boiling point of water, which is also called hyperthermophilic, such as the Cephalobus and Acidianus pyrolobus. Some of the Eukaryote can live into the frigid waters. For the example, those microorganisms are lived into the Antarctic. Next is the Urakuta. So Urakuta is also known as the Eukaryote, which inhibit the extreme environments of one or the other. So some of them are extremely halophiles, and which means that they can alive or inhibit highly saline environments. An example of extremely halophile is the halobacterium. Some of them help into the methane production, and they are called the methane producing bacteria, such as methanobacterium and methanosarcina. Some Eukaryote or Eukaryote can inhibit the extremely acidic environments. They are called acidophilic, such as the thermoplasma and ferroplasma. And next is to the Picrophilus. Third is the Eukaryote. This phyla is not yet officially recognized as taxonomically, but Eukaryote contains those organisms that are discovered from a single hot spring obsidian pool present into the Yellowstone National Park, USA, and the mixed laboratory cultures of the Eukaryote indicate that they are hyperthermophilic. So next domain is the bacteria. Is the domain bacteria, which is also called as Eubacteria. These are also prokaryotic cells with bacterial ribosomal RNA and contain the diacylglycerol diester lipids in their membrane. They are also called Eubacteria or true bacteria. Their cell membrane contains the ester linkage between the unbranched fatty acid chains and glycerol. Their cell wall made up of the peptidoglycan. There are present 5 phyla of the bacterial domain, such as the Proteobacteria. The example of Proteobacteria is the E. coli and Salmonella typhus. Next is the Legionella. And the next is the Helicobacter pylori, and the next is it is the cause of the many ulcers. Next is the Neisseria gonorrhea, cause of the gonorrhea. Next is Cyanobacteria. The example of Cyanobacteria is the photosynthetic blue-green bacteria which produces the oxygen gas. Next is Eukaryote. The example of Eukaryote is the Clostridium, which is also called as the tetanus botulism. And next is the Bacillus mycoplasma, which causes the walking pneumonia. Next is the Chlamydias. The example of the Chlamydias is the Giardia or Jarda and Chlamydia, STD, etc. Next is Spirochetes or Spirochetes. The example of Spirochetes is spiral bacteria that causes the syphilis and Lyme disease. These are prokaryotic cells. They contain a nucleus without any membrane. They are also known as Eubacteria or true bacteria. Their membrane contains diacyl, glycerol, diester lipids. So all bacteria are not pathogenic. Some of them play the beneficial roles by producing the antibiotics in food. They contain ester linkages in their membranes. This linkage is formed between unbranched fatty acid chains and glycerol. Unlike the Archaea and Eukarya, the bacteria's cell wall contains peptidoglycan. They are sensitive to all antibacterial drugs, but they are resistant to most of the antibiotics that affect the Eukarya. They contain a unique bacterial ribosomal RNA, which is distinctly different from the ribosomal RNA of Archaea and Eukarya. Next I am going to discuss about the phyla of the bacteria. First is the Proteobacteria. It includes all the enteric bacteria such as the E. coli and Salmonella, typhus, Legionella and Helicobacter pylori, which is the cause of many ulcers, and Neisseria gonorrhea, which is the cause of the gonorrhea. These are closely related to eukaryotic mitochondria. Next is Cyanobacteria. These include all photosynthetic blue-green bacteria which produces oxygen gas, and Eukarya that includes the Clostridium, tetanus, botulism and Bacillus mycoplasma, which causes the walking pneumonia. Next is the Chlamydias. These cause the parasites, and these include parasites which causes the Giardia or Giardia and Chlamydia STD. Next is the Spirochetes and Spirochetes. These include spiral bacteria that causes the syphilis and Lyme disease. So next I am going to discuss about the domain Eukarya. These are eukaryotic cells with a membrane-bound nucleus. Their membranes contain membranes between the unbranched fatty acid chains and glycerol. They lack the peptidoglycans on their cell wall. Eukarya inhibit the antibacterial antibiotics but sensitive to antibiotics that affect eukaryotic cells. There are presently 4 kingdoms of the Eukarya such as Protista. These are slime molds, euglenoids, algae and protozoans. Fungi, these are sac fungi, club fungi, yeast and molds. Planty, these are mosses, ferns, conifers and flowering plants. Animalia, these are sponges, worms, insects and vertebrates. These are all eukaryotic cells. Eukaryota and Eukarya contains a membrane-bound nucleus. Eukarya contain the ester linkages in their membranes. This linkage is formed between unbranched fatty acids and fatty acid chains and glycerol. The cell wall of Eukarya contains no peptidoglycan. They can inhibit the antibacterial antibiotics but sensitive to antibiotics that affect the eukaryotic cells. Next is the kingdoms of Eukarya. So Eukarya has 4 kingdoms such as Protista. These are single-celled eukaryotes found on land, in water, or living inside the other organisms. The Protista includes Euglena, amoeba, and paramecium. Next is fungi. These are unicellular or multicellular organisms with the eukaryotic cell types. Their mode of nutrition is absorption, and the fungi include mushrooms, bread molds, water molds, and yeast, etc. Next is the plantae. They are multicellular eukaryotic cells. The plantae includes flowering plants, gymnosperms, conifers, ferns, mosses, etc. Next is Animalia. Animalia are multicellular and eukaryotic cells. These include sponges, jellyfish, corals, fish, worms, insects, and vertebrates. Next I am going to discuss about the current system. As the scientists learn more about the organisms, classification systems changes. So the genetic sequencing has given the researchers a whole new way of analyzing the relationships between the organisms. The current 3-domain system groups organisms primarily based on differences in the ribosomal RNA structure. Ribosomal RNA is a molecular building block for ribosomes. Under this system, the organisms are classified into the 3 domains and 6 kingdoms. The domains are Archaea Bacteria, Eukarya. The kingdoms are Archaea Bacteria, Ancient Bacteria, Eukarya, True Bacteria, Protista, Fungi, Plantae, and Animalia. First is the Archaea Domain. This Archaea Domain contains single-celled organisms. Archaea have genes that are similar to both bacteria and eukaryotes. Because they are very similar to bacteria in appearance, and they were originally mistaken for bacteria. Like bacteria, Archaea are prokaryotic organisms and do not have a membrane-bound nucleus. They also lack internal cell organelles, and many are about the same size and similar in shape to bacteria. Archaea reproduce by binary fission and have one circular chromosome and use flagella to move around in their environment as do bacteria. Archaea are different from the bacteria in their cell wall composition and differ from both bacteria and eukaryotes in membrane composition and ribosomal RNA type. These differences are substantial enough to warrant that the Archaea have a separate domain. Archaea are extreme organisms that live under some of the most extreme environmental conditions, and this includes within the hydrothermal vents, acidic springs, and under Arctic ice. Archaea are divided into 3 main phyla, which are the Eukaryote, and Urakuta, and Eukaryote. First is the Eukaryote. Include many organisms that are hyperthermophiles and thermoacidophiles. These Archaea thrive into the environments with great temperature extremes. Hyperthermophiles and in extreme hot and acidic environments like the thermoacidophiles. Archaea known as methanogens and are of Eukaryote phylum. They reproduce methane as a byproduct of metabolism and require an oxygen-free environment. Little is known about the Eukaryote. Archaea, as few species, have been found living in places such as hot springs, hydrothermal vents, and obsidian pools. Next is Bacteria Domain. So bacteria are classified under the bacterial domain. These organisms are generally feared because some are pathogenic and capable of causing the disease. However, bacteria are essential to life, for some are part of human microbiota. These bacteria perform the vital functions such as enabling us to properly digest and absorb nutrients from the foods we eat. Bacteria that live on the skin prevent the pathogenic microbes from colonizing the area and also aid in the activation of the immune system. Bacteria are also important for the recycling of nutrients in the global ecosystem, as they are primary decomposers. Bacteria have a unique cell wall composition or ribosomal RNA type. They are grouped into the 5 main categories. First is the Proteobacteria. This phylum contains the largest group of bacteria and includes the E. coli, Salmonella, and Helicobacter pylori. and Vibrobacteria. Next is Cyanobacteria. These bacteria are capable of photosynthesis. They are also known as blue-green algae because of their color. Next is the Firmicutes. These gram-positive bacteria includes the Clostridium, Bacillus and Mycoplasmas, the bacteria without cell walls. Next is the Chlamydiae. These parasitic bacteria reproduce inside their host cell. Organisms include Chlamydia. Trachomatis causes the chlamydia STD and Chlamydiophila pneumoniae causes the pneumonia. Next is Spirochetes. These corkscrew-shaped bacteria exhibit a unique twisting motion. The examples include the Borrelia, Burgdorferi, which causes the Lyme disease, and Treponema pallidum causes the syphilis. Next is the Eukaryote Domain. The Eukaryote Domain includes the eukaryotes or organisms that have membrane-bound nucleus. This domain is further divided into the subkingdoms, which are Protista, Fungi, Planty, and Animalia. Eukaryotes have ribosomal RNA that is distinct from the bacteria, and archaeans, plant and fungi organisms contain the cell wall that are different in composition than bacteria. Eukaryotic cells are typically resistant to antibacterial antibiotics. Organisms in this domain include the protists, fungi, plants, and animals. So examples include algae, amoeba, fungi, molds, yeast, ferns, mosses, flowering plants, sponges, insects, and mammals. Comparison of classification systems. Systems for classifying organisms change with new discoveries made over time. The earliest system recognized only two kingdoms, plant and animal. The current three-domain system is the best organizational system we have now. But as new information is gained, a different system for classifying organisms may later be developed. Here is how the five-kingdom system compares to the three-domain system, which has the six kingdoms. Five-kingdom system is Monera, Protista, Fungi, Planty, and Animalia. So this is the comparison of classification system. First is the Archaea Domain, which includes Archaea Bacteria Kingdom, Bacteria Domains, which include Eukarya Kingdom, Eukarya Domain, which includes Protista Kingdom, Fungi Kingdom, Planty Kingdom, and Animalia Kingdom. So next is the classification of life, the three-domain concept. A domain is the highest taxonomic level of species in biology. In 1977, Woese and his co-workers developed the groundbreaking three-domain system. They proposed this classification based on differences in the sequences of nucleotides in the cell's ribosomal RNAs, known as the 16 subunit ribosomal RNA. They created the first worldwide tree of biological life by comparing the 16 subunit ribosomal RNA nucleotide sequences. Similarities and differences in ribosomal RNA nucleotide sequences reveal how closely or distantly related different cells and organisms are. Comparing nucleotide sequences in 16 subunit ribosomal RNA from the different cell types reveal the three unique cell groups. They are the Archaea, the Bacteria, the Eukarya. So, according to the LUCA, and LUCA stands for the last universal common ancestor. There are three domains. So first domain is Eukarya, then comes domain Archaea, and then comes domain Bacteria. LUCA and the tree of life. This classification based on the 16 subunit ribosomal RNA provided the most robust possible support for the Darwin's common ancestry concept. Hence it became the fact that all the cells came from a common ancestor. Cell termed as the last universal common ancestor, LUCA. We still know little about how the LUCA used to live and look like. Before Woese, scientists believed that there are only two types of life, Eukarya and Prokarya. So this is the common ancestor, which includes three domains. So first domain is the bacteria, which includes green non-sulfur bacteria, gram-positives, purple bacteria, Cyanobacteria. Next is Flavobacteria and Thermophiles. Next domain is Archaea, which includes the Halophiles and Urakuta, Methanosarcina, Methanobacterium, Methanococcus, T. cellular, and this is the Eukaryote, which includes the Thermophiles and Pyrodicticum. Next is the domain Eukarya, which includes animals, fungi, plants, ciliates, flagellates, trichomonads and microsporidia, diplomonads, slime molds and entamoeba. Now the question, why the Archaea and Eukarya are together? Was believed that Archaea and bacteria are molecularly distinct despite their similarities, and it should form the separate domains on the evolutionary tree. Strikingly, evolutionary biologists place the LUCA between bacteria on one side and Archaea on Eukarya as sister lineages on the other side. They proposed that the domains of Archaea and Eukarya emerged from a common archaeal eukaryotic ancestor that emerged from a member of the domain bacteria. So we can say in the beginning that the tree of life had only two branches, which are Archaea and bacteria. Now the question, why the bacteria and Eukarya are not together? Archaea were once regarded to be the most primitive prokaryotic group. On the other hand, the bacteria were assumed to be closer to eukaryotes. Studies of ribosomal RNA indicate that it is not the case. Instead, the Archaea seem to be related to eukaryotes more closely than bacteria. So Archaea resembles bacteria in their apparatus for metabolism and energy conversion. But in the case of molecular level, the methods of the handling the genetic information like the replication, transcription, translation, Archaea resembles the eukaryotes more closely than bacteria. This finding emphasizes that the similarity of cellular organisms and common ancestry are two different things. Now I am going to discuss the domain Archaea. So Archaea is a group of single-celled prokaryotic organisms that have distinct molecular characteristics than bacteria. Archaea comes from the Greek word archaeos, meaning ancient or primitive. Some Archaea indeed exhibit the characteristics worthy of that name. The best known Archaea are species that live in highly inhospitable environments, like they are referred to as the extremophiles. Archaea are often found inhabiting environments that we humans avoid, such as the bogs and sewage treatment plants, ocean depths and salt brines, and hot acid springs. However, they are widely spread in the less extreme and more homely environments, from soils and lakes to the stomachs of cattle. This is the domain Archaea shown into the figure. Domain Archaea includes some major groups which are methanogens, strict anaerobes that produce the methane CH4 from carbon dioxide and hydrogen. For example, Methanosarcina, Barkery, and Brevibacter, Simidiae, and Methanosarcina acetovorans, Methanococcus mariplodis, etc. Next is halophiles. So halophiles live in extremely salty environments such as the Dead Sea or certain deep sea brine pools that possesses a salinity equivalent to 5 mgCl2 magnesium chloride. For example, Halobacterium salinarum. Next is the Halophyrix volcanii. Next is Dunaliala, Salina, and next is the Tetragenococcus halophilus, etc. Acidophiles are acid-loving prokaryotes that thrive at the pH as low as 0, and such as that found into the drainage fluids of abandoned mine shafts. For example is the Acidithiobacillus Phaeroxidans. Next is Acidithiobacillus caldus, Acetobacter aceti, and Pediococcus acidilactici, etc. Next is thermophiles, the archaea that live at very high temperature, like the hyperthermophiles or thermophiles that live in the hydrothermal vents on the ocean floor. For example, as the Alicyclobacillus Alicyclobacillus and Acidoladarius and Alicyclobacillus acidoterrestrus, Alicyclobacillus acidophilus and Alicyclobacillus tolerans, etc. Next is the domain bacteria. So bacteria are microscopic organisms with only one cell. Their cellular structure is simpler than the other organisms. So bacteria lack a nucleus or membrane-bound organelles. Instead, a single DNA loop stores its genetic code. A plasmid is an additional circular genetic material found in some bacteria. The plasmid often contains genes that give the bacteria an edge over the others. It could, for example, contain a gene that makes the bacteria resistant to a certain drug. Bacterial cells come in a variety of forms. So bacteria are classified into 5 groups according to their basic shapes. So first shape is the spherical, which is also called cocci. Next is the rod shape, which is also called bacilli or bacilli. Next is spiral shaped, which is also called spirula. Comma, which is called as the vibrios or corkscrew, which is also called spirochetes or spirochetes. They exist as single cells in pairs, chains or clusters. Bacteria can be found in almost any environment on Earth, from the permanent ice shelf of the Antarctic to the driest African deserts, even to the internal confines of plants and animals. Some organisms such as plants and animals and humans live in or on other organisms. Bacteria have even been detected in rock layers situated several kilometers beneath the Earth's surface. In the human body, there are approximately 10 times as many bacterial cells as human cells. So bacteria exist in the soil or on decaying plant debris, where they play an essential role in recycling nutrients. So some species degrade food and harm the crops, while the others are essential in the production of fermented foods such as tea, yogurt and soy sauce. Only a small percentage of bacteria are parasites or pathogens that infect the animals and plants. This is the domain bacteria. Different shapes of bacteria are shown into the figure. Next is the cyanobacteria. The most complex bacteria are cyanobacteria. Cyanobacteria contain elaborate arrays of the cytoplasmic membrane. The membrane serves as the site of photosynthesis. The membranes of cyanobacteria are very similar to the photosynthetic membranes present within the chloroplast of plant cells. Like eukaryotic plants, they can produce oxygen by splitting water. Many cyanobacteria are also capable of nitrogen fixation. They can convert the nitrogen gas into a reduced form of nitrogen such as ammonia. Cells then use this reduced nitrogen to synthesize the nitrogen containing organic compounds, for instance amino acids and nucleotides. Those species capable of both photosynthesis and nitrogen fixation can survive on the barest of the resources. Examples of cyanobacteria are nostoc, oscillatoria, spirulina, microcystis and anabina, etc. This is cyanobacteria as shown into the figure. So it includes the mucoid sheath. This is mucoid sheath. This is capsule. And this is slime coat. This is carboxysome, this is ribosome, and this is nucleoid or DNA. This is thylakoid. Thylakoid contains the phycobilisome and thylakoid membrane, and the cell wall contains the cell membrane. This is cell membrane, and this is peptidoglycan layer, and this is the outer membrane as shown into the figure. Next I am going to discuss about the actinomycetes. These are branching filamentous bacteria. They are termed actinomycetes. Actis mean ray and myx mean fungus, because they resemble the radiating rays of sun when seen in tissue lesions. Examples of the actinomycetes is bifidobacterials, mycobacterials, and micrococcales, and this is propionibacterials and actinomycetia, etc. This is actinomycetes as shown into the figure. Next I am going to discuss about the mycoplasmas. They lack a cell wall, thus lack a fixed morphology. They have round or oval bodies with interlacing filaments. Examples of mycoplasma are Mycoplasma genitalium, Mycoplasma hominis, Mycoplasma pneumoniae, Mycoplasma bovis, Mycoplasma mycoides, etc. This is mycoplasma, and so this is the lipoprotein membrane, and this is soluble protein. This is ribosome in green color, this is DNA in black color, and this is soluble RNA in white color as shown into the figure. Next I am going to discuss about the domain Eukarya, coming from the Greek words eu, which means true, and karyon which means nut. The domain Eukarya is composed of organisms having a true nucleus. The domain Eukarya consists of organisms with a true nucleus and membrane-bound organelles. This domain includes all animals, plants, and fungi. Humans are eukaryotes, as are falcons and chimpanzees, hydras, and mushrooms. So Eukarya also includes some much smaller things, for instance yeast, amoebas, and algae. This incredibly diverse group of these species is solely linked by the fact that they have similar types of cells. All of their cells have certain features that differentiate them from the prokaryotic cells of bacteria and archaea. Eukaryotic cells perhaps contain the most complex external and internal structures and physiological and reproductive processes. Among all the domains in the biological world, members of domain Eukarya have the most significant body size and body mass. There are some single-celled eukaryotes such as the protists and fungi. Others are multicellular. The examples of eukaryotes are animals, fungus, protozoa, plant, dinosaur, diatom, ciliate, brown algae, etc. Microbial genes are now known to transfer not only vertical from a parent organism to its progeny, but also horizontally to the distant cousins, such as other species and other genera. As a result, the old three-branch tree of life model has evolved into a net of life. This is a eukaryotic cell as shown into the figure. These are eukaryotes as shown into the figure. Not included in the three-domain system, neither of these approaches included viruses or prions. In the classification scheme of life, many scientists do not consider these things to be living, as viruses hijack a cell's machinery to replicate itself, while the prions are misfolded proteins which somehow use the other proteins to fold incorrectly. Both of these genetically based molecules have a mean for reproduction, which is why many scientists consider them living for classification purposes. Several alternate theories, including these groups into domain system, have been proposed, often creating new domains for each group. Next, I am going to discuss the future of classification. The three-domain system is unlikely to be final word on the classification. The system is based on the current state of knowledge. As knowledge increases, the three-domain system may need revision. For example, the number of domains may change as scientists learn more about those life forms we currently know little about. A recent discovery illustrates this point. In 2003, scientists identified a new virus which is called mimivirus. It resembles the bacteria in size and number of genes. However, the virus cannot respond to stimuli or grow by cell division, both of which are traits of bacteria and other living organisms. Mimivirus' unique combination of traits seems to place it at the boundary between the living and non-living things. So some scientists think mimivirus might represent a new domain of life. Next, I am going to discuss about the difference between the kingdom and domain. The key difference between the kingdom and domain is that the kingdom is one of the 5 major groups of living organisms, while the domain is one of the 3 taxonomic categories of living organisms. Above the kingdom level, there were many attempts of classification of organisms on Earth. Till 1977, kingdom system was universally accepted across the world, starting from the 2 kingdom systems called the Linnean system way back in 1758, when the life forms were divided into plants and animals. The world has come to recognize the 3-domain system as the most current and most scientific system of classification of organisms. Though there are some similarities, the kingdom system of classification of organisms has now been relegated to having the relatively little significance since the 3-domain system popularized in all parts of the world. The aim of this article is to find out the difference between the kingdom and domain system of classification of organisms. Now the question is, what is kingdom? The old 2 kingdom system of classification comprised of 2 kingdoms, plant kingdom and animal kingdom. A Linnean system called all the organisms that move with the anima, with a soul, and fungi got classified as plants. The system went on adding the kingdoms. However, the 5 kingdom classification system was believed to be the complete system after the discovery of bacteria, algae and electron microscope. Hence, the 5 kingdom system proposed in 1969 is the most updated system of classification. Today, even though the 6 kingdom classification is more accurate. Planty, Archaea Bacteria, Eukaryote, Protista, Animalia and Fungi are the 6 kingdom included in the 6 kingdom classification. In this classification system, the 5th kingdom, Monera, has been divided into Archaea Bacteria and Eukaryote, thus bringing the number of kingdoms to 6. The 5 kingdom classification includes the 5 kingdoms, namely Monera, Protista, Fungi, Planty and Animalia. In all the classifications, whether the 5 or 6 kingdoms, a kingdom has subdivisions such as phyla or divisions. In fact, kingdom Animalia contains of phyla, while the kingdom Planty has divisions. What is domain? 2 scientists, Woese and Wolf, in 1977 completely changed the system of classification of organisms in the world when they made the use of 165 ribosomal RNA sequences. Till 1977, the world had to come to accept the bifurcation of life forms into eukaryotes and prokaryotes. But it was the discovery of Archaea, the microorganism that could live without oxygen, so reminded one of the ancient environment of Earth that was devoid of oxygen, that forced the scientists to conceive of a 3rd category of organisms. Therefore, Archaea was accepted as being different from the earlier 2 classifications, and thus the 3-domain system came into existence. Accordingly, the 3 domains include the bacteria, Archaea and Eukarya. So now the question is, what are the similarities between the kingdom and domain? Kingdom and domain are the 2 types of categories to classify the living organisms. Kingdoms are coming under the domains, and all the living organisms belong to 3 domains as well as to 5 kingdoms. Now the question, what is the difference between the kingdom and domain? The 3 domains and 5 kingdoms are 2 types of classification system to characterize the living organisms. The key difference between the kingdom and domain is that the kingdom is one of the 5 major groups of living organisms, while the domain is one of the 3 taxonomic categories of living organisms above the kingdom level. Thus, domain is a category above the kingdom level. Accordingly, there are 3 domains, namely Bacteria, Archaea, Eukarya. On the other hand, the kingdom is a major category of living organisms below the domain level. There are 5 kingdoms, which are namely Monera, Protista, Fungi, Planty, and Animalia. Hence, this is another difference between kingdom and domain. Kingdom versus domain as shown into this table. So, first is the definition. The definition of kingdom is one of the 5 categories of living organisms. Domain is the one of the 3 major categories above the kingdom level. That includes the living organisms. The categories of the kingdom are 5, which are Monera, Protista, Fungi, Planty, and Animalia. And the categories of domain are 3, which are Bacteria, Archaea, and Eukarya. The level of the kingdom is below the domain level. The level of the domain is always above the kingdom level. If you like my video, please subscribe to my YouTube channel and press the bell icon as well for further notifications. Thanks for watching. Thank you so much for your time.